cardiomyopathy or heart failure is a leading cause of the death in a human patient. Half of the patients with heart failure is dilated cardiomyopathy. And another half of the patient with heart failure normally is with hypertrophic cardiomyopathy. So for the dilated cardiomyopathy, there's drugs or therapeutic option available. But for the hypertrophic cardiomyopathy, so far there's no therapeutic option available. My name is Wei Guo. I'm a muscle biologist. I'm a faculty member in animal science. My research interest is mostly focused on muscle structure and function. Mainly I will focus on cardiac muscle and skeletal muscle. The muscle has a special structure, so we define this unit as a sarcomere, which is the smallest contractile unit. So in this model, I only choose 3 major proteins, which is called actin, myosin, and titin. So these 3 proteins are taking up about 80% of whole muscle mass. And this basic unit can repeat many times. And then the repeats can connect each other. And that will form a long cylindrical rod, which we call this a myofibril. There are about 500 to 1,000 myofibrils in one muscle cell. That takes up about 80% of the space in the muscle cell. It's much like optical fiber cable filled with glass fibers. So mainly our research focus is focused on one protein which is called titin. So why we focus on this protein, that's because titin is like a rubber band. It's a spring-like protein, which means this protein can be stretched and released. And when you stretch this protein will produce passive force. So this elastic property enables this protein to be stretched and prevent muscle damage from overstretching. If you overstretch, so the muscle structure will be broken, and then the muscle falls apart. You got the muscle injury or muscle damage. So we know the long rubber band can be stretched easily, which is the long rubber band produces less passive stiffness or passive force, versus the shorter one. So you can see the shorter one is stretched a little bit harder to stretch because the shorter one can produce more force. So the basic idea is the titin as a spring-like protein can be expressed naturally in our body, be long and short 2 forms. So that's amazing. So if you can imagine in a normal heart tissue, the short titin isoform takes up about 60% in a normal healthy heart, 60% of short titin isoform. But if the short isoform expressed more, so that means the heart will be more stiffer. So which can be commonly seen in the patient with hypertrophic cardiomyopathy. And if the higher level if the short isoform expressed less in the heart muscle, you can imagine the heart will be more compliant. So this disease condition can be seen in a patient with dilated cardiomyopathy. So one of our major research is focusing on to understand how the long and short titin isoform are regulated. And ultimately, we are trying to develop a therapeutic strategy to adjust the muscle stiffness through correction the long and the short titin isoform ratio to treat the hypertrophic cardiomyopathy.